how different types of microbes will have different levels of resistance to methods of disinfection and sterilization. So we'll pick up on that. There's still a couple slides sort of in that vein before we talk about physical methods of microbial control. Okay, so we're going to talk through a couple of these different like prokaryotic targets and how they work, but this is pretty intuitive stuff, so we can go through it, I think, hopefully without too much confusion. So we'll kind of start our cells out on the outside of the bacterial cell with a cell wall. Some of these disinfectants will target the cell wall, and then the cell membrane. I just put on the same slide, but starting with the cell wall first, some disinfectants and sterilants will target synthesis of those cell wall components, particularly some antibiotics we'll talk about later. But we hit this in block 1. Where are those cell wall components actually made? Where are they synthesized? So proteins will be made in the cytoplasm, but they're actually translated pretty close to like the cell membrane itself. And then they go into the cell membrane, and they get kind of prefabricated, if you will. So all those peptidoglycan structures will get made in cytoplasm, and then pushed out with carrier proteins up into the cell wall. Okay. So if you disrupt cell wall synthesis, yes, it will mess up the cell wall, but it's actually occurring in the cytoplasmic membrane. That's where the production of those things are. You also have other agents that will impact cell wall integrity directly. That will cause the cell to burst. Those include oftentimes things like detergents and alcohols. With the cell membrane, what this diagram is showing us are molecules called surfactants, which is an abbreviated compound word for surface activating agents, surfactants. And what those things do is kind of integrate into that membrane, which in small doses might cause like a little bit of destabilization, maybe some leakage of bacterial cells. But in high enough doses, this will just completely destroy the integrity of that membrane, and it'll end up killing the bacterial cell. So also a loss of functionality, and we'll lose that permeabilization. We also have other agents that can interfere with the transport functions too, which would probably be less likely to be bactericidal. And then the remaining 3 targets are all on this one slide, but proteins, enzymes, which are types of proteins, and then nucleic acids. We have some disinfectants like alcohols, aldehydes, and heavy metals, which could disrupt protein function by essentially causing them to denature, losing their tertiary structure, their quaternary structure. And some classes of antibiotics that, again, we'll go over later today, that will target protein synthesis. So we're talking about the bacterial ribosome. Those target the bacterial ribosome selectively. Other agents will target nucleic acids. Either their synthesis, so they'll interfere with DNA replication, or they'll interfere with transcription, like RNA synthesis. Or they'll do something to directly damage the nucleic acids, the DNA or the RNA themselves. And those we briefly talked about when we were talking about mutations as well, with the spontaneous mutations. Alkylating agents, exposure to radiation, things like that can damage nucleic acids. So with these enzymes, we're talking about things that will oxidize enzymes and produce free radicals from those enzymes, destroy their functionality. Heavy metals would be things like copper and silver that can have antimicrobial functions as well. And in this slide, it's sort of in the reverse, talks about those different categories of different disinfectants. Not talking about antibiotics in this slide, but just the disinfectants and sterilants themselves, and sort of what they do to damage microbial targets. And we have a little cartoon of a cell, which has a lot of details, so you do not need to memorize anything that's on this cell diagram here. It's just meant to kind of show you where in that bacterial cell these things will be targeting. Okay, and then lastly, we'll circle back to our old friend, the soap molecule, the mighty soap molecule, how this thing works on a molecular method. A soap molecule is kind of a cool structure. It is considered amphipathic, which which means that on one end we have a hydrophilic component. So you have an ionic head here, and then you have a very long chain of hydrophobic parts of this molecule, which will make it resistant to interacting with water. Okay. So what happens is when you get in contact with a germ, which they tend to be, greasy, I guess, in nature, those hydrophobic parts, that hydrophobic table, will bind to the germ particle in the same manner that it would bind to dirt or grease. And then the hydrophilic heads of those soap molecules will interact with water and essentially form this circular structure or spherical structure around the microbe or the piece of dirt, whatever, so that when you have water applied, it just lifts that germ right off the surface. So that's how soap works. When you have the mechanical action of you like rubbing your hands together when you're washing your hands, that helps facilitate the removal of those dirt particles and germs from your hands. And that's why if you're just rinsing your hands with water briefly, that's really not going to be as effective. Okay, so we have another cartoon that's showing how soap molecules work to lift dirt off of surfaces, but also there's another mechanism that works similarly with some particular pathogens like envelope viruses. So this figure is showing an envelope virus like a coronavirus, and these amphipathic soap molecules directly integrate into that envelope and basically act like a surfactant. So they weaken the integrity of that lipid envelope and cause it to lose its structural integrity, basically just pull this thing apart. Okay, so the next couple slides we'll talk about some physical control methods. We've talked about these a little bit already, but we would use these when we're not able to chemically disinfect when it's impractical or otherwise just undesirable. Probably the most tried and true way to kill pathogens is to apply excessive amounts of heat to them. So we'll talk about a few methods there, but what heat does ultimately, at least at low levels, is it starts to denature proteins and nucleic acids. The cold doesn't generally kill, it's not generally considered to be microbicidal. So if you want to, for example, slow down microbial growth, you can stick it in like a 40-degree refrigerator, and that will slow down the metabolism of those bacterial cells. You can even freeze bacteria and viruses. Sometimes they will form crystals when you freeze them at like -20 or -80. So if you want to preserve them, you can add a cryopreservative like DMSO or something to prevent the crystal formation. But in general, this is not, like low temperatures are not a way to get rid of microbes, they're a way to preserve them for further study and things like that. Okay, so going back to killing, our favorite large-scale method is the autoclave. A lot of people in this room have probably heard of autoclaves before, worked in hospitals where they use autoclaves. Generally speaking, autoclaves that are in hospitals are pretty big pieces of equipment. You can use them for high throughput work. Ideally, you have autoclaves for doing cleaning work, like if you're making media or you're trying to sterilize equipment. And then you have different separate autoclaves that you use for garbage, for trash that needs to be sterilized. Sometimes you only have one and you have to use them for the same purpose. They are considered to be sterilizing pieces of equipment. We'll talk about the conditions here in a second, but that is great. The only problem with autoclaves is oftentimes these things get damaged and it's very hard to fix them. In hospitals, I can't tell you how many times we have like an autoclave go down and then you have to make arrangements to ship your waste out someplace and import media in. So they're complex and they do require a lot of maintenance work. No, not necessarily. So this is a fairly complex piece of machinery. Some of them take up like an entire room, some of the larger autoclaves. And some of them, like the one I was telling you about yesterday, like the one we have with the amyl, is the size of a big canister. It looks like R2-D2, like small little guy, low capacity. But most of them are pretty big. So you load a tray on there with all of your stuff. And then you have like a water reservoir that's used to heat up water to boiling temperatures. And then you have a jacket or a sleeve on the inside of this chamber here. This door is an interlocking door. It's a very tight seal. So when you turn on a cycle, program cycle, it'll start boiling the water in the water reservoir. And that steam that's produced will start coming down, usually to the top. So the steam is lighter than the rest of the air. And the air will get forced out of the autoclave through these ducts in the bottom. So the air gets forced out, and you should have pretty good coverage of steam in that chamber. With the temperatures, 121 degrees Celsius, that's about 250 degrees Fahrenheit or so. What's the temperature that water boils? Fahrenheit? 212. So you have well above boiling temperatures in there. And you're supposed to be able to use the same for liquids, yeah? So how might you be able to sterilize a thermal stable liquid if you have to have temperatures around 250 degrees Fahrenheit? Pressure, yeah. So you'd have to increase the pressure in there. And that's what that tight seal does when you close that door. It increases the pressure to around, I think 15 to 20 psi above atmospheric pressure. So because of that, you can get both high temperatures and you can have high pressure. And then you get good penetration of the steam to all the surfaces that you need to decontaminate in that autoclave. Because of that, particularly at the higher temperature range, you can adjust the settings as needed, do a little bit longer, a little higher temperature. But because of that, this is rated as a sterilizing piece of equipment, including spores, so it should be sporicidal as well. If you have to confirm whether or not your settings have successfully killed all the spores, if it's important for your work, there are ways to do that. There are spore strips you can put in that autoclave that have viable spores from some non-pathogenic spore-forming bacteria. Geobacillus thermophilus is a big one. And then you just take that spore strip off and you incubate it at high temperatures. And if it germinates, then you fail to induce sporicidal conditions. But anyway, point is, this is a good piece of equipment, but it is expensive, has a lot of maintenance requirements, it goes down, there are problems associated with that.
Do you have to clean it?
Probably, that's probably part of the routine maintenance. I never had to do that as on a user level, though. But it does require, I think, annual maintenance, maybe semi-annual maintenance. And that might be part of the process. I've seen some dirty ones too.
E40s.
Yes, sir.
Medicals
Oh, what?
are always busy. Big ones.
What's it?
Refrigerators.
Oh, yeah. So that's kind of basically what this thing is a pressure cooker.
Put
Yeah,
together
except for it's
completely.
designed specifically for, using to disinfect medical equipment and stuff like that. But yeah, that's the same principle as a pressure cooker. Okay, limitations. What would be some examples of some heat-sensitive materials that you don't think would be able to withstand 250 degrees Fahrenheit and heavy amounts of steam? Yeah, a lot of things. Rubbers, plastics, electronic equipments, a lot of endoscopic materials perhaps wouldn't do well, anything with optics. So there are a lot of limitations on this. Thermal labile materials. What does labile mean? Susceptible to destruction usually. So thermal stable would be okay, it can tolerate that. If it's thermal labile, it cannot tolerate those high temperatures. So you can't put thermal labile things like vaccines through an autoclave. Yeah, and then if you don't, we talked about this before with bioburdens and biofilms on particularly surgical instruments that have to be reused. You gotta get rid of those. This here should be on your slide, but there was a story that broke, probably dating all the way back to 2016, I think, over at AMSI. Kind of a scandal, and then it resurfaced again in 2023. But the scandal was that some surgical instruments were being autoclaved, and they came out of the autoclave with tissue still on those instruments. And as a result, they had to shut down 50% of their operating rooms. They had to delay and reschedule some surgeries, some of which were important surgeries. So really bad stuff. And I think somebody working at the hospital reported it to the local paper, the News Gazette or whatever, San Antonio. So fast forward like another 6 or 7 years in 2023, the same thing happened again. second time an incident, and then Joaquin Castro, who's the U.S. representative for the 20th Congressional District in Texas, which covers San Antonio, wrote a big nasty letter to the commander of BAMSI saying, why is this still a problem? This should not be an issue. So, but yeah, it all just related back to the fact that somehow their SOPs for decontaminating surgical equipment did not ensure that they were rinsing off the bioburden before they went into the autoclave. Was anyone at BAMSI, just out of curiosity? Anybody been stationed there before? Oh, yeah, thank you, John. Okay. Yeah, were you aware of that at all? Did you hear about that story?
Yeah, I was there for the first two, not the second.
Okay. Oh, wow. Okay. Yeah, I should pick your brain about that at some time. I'm curious. Okay, so we talked about autoclaves and the fact that you're using steam heat to disinfect the penetrating hard steam. You can also use these things. These are more like analogous to ovens, dry heat. With dry heat sterilization, you do need higher temperatures, usually for longer periods of time, which means that the trade-off is that you have more restrictions on what types of things can go in there probably. So this is not at higher atmospheric or anything, or it's not at increased pressure. But you do have them, and they're typically a little bit simpler and easier to use than autoclaves. Another method is pasteurization. This doesn't really relate to medical stuff so much, but it's a good example of a liquid process, a process to reduce microbial loads in liquids. Usually milk, so this is more useful in the food industry, like you can see here, the start of the pasteurization process. But with pasteurization, you are using, again, heat setting about 63 degrees centigrade or 145 degrees Fahrenheit for 30 minutes. And what that does is drastically and dramatically reduce the microbial load in that liquid. But it's not a sterilization, so I probably should have had this header be just heat instead of sterilization. Eventually, pasteurized milk will go bad, yeah. So it does have a short shelf life, and that's because the microbial load has been reduced and not eliminated. You can also do ultra-high temperature pasteurization, kick that up to a higher temperature, and get further reduction of microbial load, which corresponds to a longer shelf life, which is great. You have a question?
Just a random question about that. So,
Yeah.
with the ultra high pasteurized milks and stuff, is there really any nutritional value left in that?
I don't know. Yeah, maybe. I don't know, like what it does to
That's
the.
not a lot. That's pretty much all that matters.
It's denatural proteins.
Yeah, where?
All of Western Africa.
Yeah. Yeah, that was my experience as well. I was stationed out in Kenya for 2 years. That was my first CONUS research and surveillance job, and I used to order it from Amazon, like bulk crates of UHT milk and all their things like that that I just survived on for long periods of time. But I don't know about the nutritional value of UHT milk, but I know that people don't like it because it does have an altered taste profile. Most of you have had UHT milk before, no? Oh, sorry. I'm from Wisconsin, so it's like, aw, sad. Has anyone ever had UHT milk, ultra high temp? Okay. Do you think it tastes different?
I did.
Yeah. Not a fan. Okay.
Is it the same as the extremely long shelf life milk that you like to give us up?
Yep. That's the same stuff, yeah. So hypersane love that, I like the taste, but it has like a burnt taste because of the Maillard reaction, right? It starts to break down the sugars, I think, some of the other proteins in milk, but yeah, it's like caramelized, partially caramelized and kind of like a burnt sweet taste to it. I think it tastes great, but I'm starting to like that. Anyway, so that's pasteurization. And then, last but not least, my favorite, incineration. I mean, we don't even have to talk about what microbial target is hit with incineration. It's reduced to ash. Super satisfying. So, we used to have a lot of incinerators out, like, hospitals oftentimes had their own incinerators associated with them, but there are probably fewer than 100 in the United States now because we have preferred methods of disposing of hazardous waste. The EPA, for example, isn't crazy about some of the smoke and byproducts from incinerators. Communities generally don't like to have large incinerators in their vicinity. People don't like it. But it is effective. And yeah, I don't know what temperatures they raise the material to, but it gets her done.
Get her done.
Oh, and then radiation. Okay, so we talked about this also a little bit. Two different types of radiation. Ionizing radiation is considerably more penetrating, higher energy radiation. Has greater penetrating power into different materials. In the US, we use some ionizing radiation in the food safety industry. Like we'll do it for meat products sometimes, and poultry, sometimes fruits, vegetables, spices, things like that. I think there's a requirement to label food that's been irradiated, but there is some sort of public misconception that irradiated food is radioactive, something like that. The public doesn't like it. But of course it isn't. It's just been radiated. So it's been shot at with gamma rays or X-rays and had all the microbes destroyed because this will destroy nucleic acids and proteins alike. Completely destroys them. And that includes spores as well. So the other type of radiation is not nearly as effective. This is ultraviolet light. It is damaging. I mean, everyone knows, right, that if you spend a lot of time out in the sun, UV light will damage your own skin. Even with your keratinized dead skin cells, it can still eventually penetrate and cause problems. But for bacteria and viruses, if non-ionizing radiation hits, it can kill them as long as it's kind of in the direct path of that ionizing radiation, of that UV light. It does not have very good penetrating power. To say. So if you're using UV light, let's say in a hood like this, if you've ever worked in a hood, you usually have like a UV lamp up here, and then obviously it's not on while you're working, but after you do another chemical disinfection of the surface when you're done, then you can turn on that UV light and just leave it on. It's not going to disinfect everything though. If areas are obscured, it's not going to penetrate and disinfect those areas, sterilize them. But this is commonly used in a lot of different applications, like water treatment facilities will pass water through UV reactors to treat water that way. And it's used in offices and other spaces as well for kind of, low-level inactivation and microbial control. And this mechanism is generally a little bit different. You're not inducing double-stranded breaks in DNA like you get with ionizing radiation. But it does cause some weird changes in DNA, like thymidine, which if you remember that structure, it is a pyridine, and it is like a single ring structure, so it lays very flat, and if you have a couple thymidines next to each other in the same sequence in DNA, what can happen is when you shoot it with UV light, those thymidine bases can start to cross-link together. So, and that's fine, it might distort the molecule a little bit, but when you need to replicate or transcribe a gene that has thymidine dimers, it's not going to work. Okay, so that can be bactericidal as well. Okay, then we have some filtration methods. This is a great option if you need to get liquids that are thermal labile, mostly clear of any contaminants. You do hear the word sterilization applied to filtered liquids sometimes, and that rarely is absolutely true. It depends on the pore size that you use to filter these things through. Some of the bigger pore sizes are about 0.45 microns long, and that will get rid of most bacteria. If you want to do finer filtration, 0.2 microns will get rid of even the small atypical bacteria. Most of them will get rid of them. But viruses, for example, those can, so viruses can escape through those pores. It depends on what the application is. If you want to go even lower than that, they do make lower pore sizes that go down to microfiltration ranges and nanofiltration ranges. Even reverse osmosis is sort of like a size exclusion type of purification. But for most intents and purposes, if you just need to clean some culture or maybe vaccine, maybe IV fluids, you can stick your liquid into a filter setup like this. This is a standard like 200 ml filter setup. It has a vacuum connection on the side there, so you can stick a vacuum to it and it will accelerate that flow rate through this pore. And then you're doing this in a hood, a sterile hood. Then when you're done filtering it through, just screw it off and you have some pretty clean media to work with. This setup is another setup where you have a filter that you can attach to a syringe for really small volumes and just squeeze that liquid through, like 5-10 mils of liquid. So, but oftentimes these membranes are like a cellulose acetate or some similar membrane like that, and you can see how it's just trapping all of those bacilli and cocci bacteria on the surface. Okay, moving on to air filtration, we have HEPA filters, high efficiency particulate air. With this concept, you're using, so instead of those pores, now you have some randomly oriented fibers, like glass and polymer fibers, you can see in that lower right-hand corner, and you're forcing air through those fibers with the intent of getting rid of particles that are greater than around 300 nanometers or 0.3 microns in size. So pretty effective at doing that. Used a lot in HVAC systems. Actually, we have a slide that talks about some of those uses for HEPA filters, but not usually going to be great for blocking small viruses. What was that lower end of virus sizes that we talked about? About 20 nanometers. Yeah, so it perhaps would not be effective for that lower end viruses. So these are some places where you would find HEPA filters being used a lot. N95 masks, most of us have probably had to wear those at some point during COVID. Not super fun, but the point here is to keep you protected from dangerous air. If you're wearing a surgical mask, it's to protect something else from your germs, from you breathing on it, right? But an N95 is supposed to be protecting your airways, so you have a close fit. You can see that nose cup around this guy's nose, nice tight fit, and it's supposed to block 95% of that particulate air from getting into your airways. If you want to bump up, a PAPR is considerably more protection. That is a powered air purifying respirator. So basically, if you can see on this guy, he's got a battery pack on the back of that, and then you have blowers attached to that. Those blowers are circulating air through HEPA filters in that battery pack. And then they send that purified air up this hose and then out in this positive pressure hood that you wear over your face. Anybody ever worn a PAPR before to work? Yeah, a bunch of you guys. So yeah, PAPRs are nice. They're less, because you have a nice hood over your face, you don't have the uncomfort or discomfort of wearing an N95. But they are noisy, right? If you've ever worn a PAPR, you have that whirring mechanical noise and then the air rushing past your ears, so that's a little distracting. And then you have to charge the batteries, of course, as well. We also have HEPA filters that work in those biological safety cabinets. You can see there on the right there. The HEPA filters would be responsible for, so let me back up a little bit. When you're working in a biological safety cabinet, the point, of course, is to kind of separate out environments. What's in the hood and what's on the outside of the hood. So you have to have airflow, like an air curtain, between this glass sash there and the bottom, which has a vent, which has air flowing through it. So that air curtain that laminar airflow should prevent air inside the hood from coming out and air on the outside from coming in. But when the air reaches this vent at the bottom, it gets recycled into this HEPA filter. And then if it's a Class 1 or Class 2 hood, it can be recirculated back into the lab. For more dangerous work, you might need a VSC3, and then it gets circulated through the ductwork outside into the environment. So there would be a duct that literally forces that air out into the environment. Oh, and then also HVAC systems usually have HEPA filters as well. So just standard building HVAC systems. Masks. I'm sorry to have to even show you this slide. You're probably sick of seeing this stuff, but it is kind of important to talk about respiratory protection and the relative types of protection that's afforded by these different categories. When you're wearing just a face mask, the highest protection, the best protection is probably that N95, because you have that woven mesh and because you have that tight fit over your face. As I mentioned before, the surgical mask's different purpose. You're not trying to protect your own airways as much as you want to save the person you're talking to from being sickened. So if you come to school and you're wearing a surgical mask, it's to protect the people around you from catching whatever you have. So reduced efficiency with those non-woven fabric layers, only 60 to 80% efficiency. If you start downgrading to stuff like this, which a lot of us, we saw a ton of during COVID, which is just like normal cloth over your face, big decrease in the efficiency of filtering out infectious particles that would be in the air, only 50 to 75%, and that depends on how you're wearing it. And just bumping back up to N95s really quick, those are best designed to be single use. So you're supposed to discard those after you wear them. Some people were wearing them for way too long. And then lastly, the old bandana around the nose thing, very inefficient, very ineffective at protecting you from microbes. Oh, and then this slide is really similar to the one we opened up this deck with when we saw that pyramid from the CDC, those different echelons or tiers of production. This is called the Swiss cheese model. And I don't know if it predates COVID, but it certainly fits in well with COVID. The idea with this model is that if you have any one single layer, no matter how good it is of protection, like you have rock solid PPE, there's still holes in that layer of protection. There's still vulnerabilities. So because of that, we layer in multiple different strategies and techniques and interventions. So even if there are multiple holes where you can get sick, like this diagram is showing the same vulnerabilities in several different intervention methods. Eventually, if you layer it with enough different protective mitigating measures, you will stop that threat. So personal responsibilities, shared responsibilities. Yeah, like most of you probably lived through this whole model before. Okay, any questions on that last bit about microbial control methods, physical control, last bit? Okay. I've got 10:35. Let's take a 10-minute break and start back up on antibiotics in 10:35.
I'm so excited about drugs.
Yes. So that was a Fusarium, right? It was a Fusarium fungal.
It's another
That was beyond my scope. That's what I was gonna ask you. Why was that so scary?
exposed to whatever.
So the particular fungus that they were smelling was a,
I'm gonna
at
get
least
to
I
it.
didn't read about it in any depth, it was like an agricultural,
Well,
it's on the agricultural.
Agroterrorism?
Agroterrorism, yeah, so this fungus was, I think it's a type of Fusarium.
there's a few. So my buddy,
It
my friend just did this one. He
destroys
has
the
like
crops.
he had
It's like an old
fractured
school
his
thing
leg.
that,
So he claimed 2 things for his med bill, and then he went to the VA and claimed the rest of his stuff. And he actually had like 270%. So he got 100% for med bill, and then he got 100% disability on top. Most of the people, they can't get that because they're retired now. Yeah, fucked up enough. Like he claimed on VA like PTSD, his fingers like all fingers like fucked up like that. Can't like squeeze tight, all that stuff. So he got 100% med bill. So he got paid for that, and then he got 100% VA disability. So he got paid for that. So he still works, but he does like random bullshit, and he's still making $50,000. VA or
I don't know how you said it.
Unless you're RFK. Oh, I see.
Shop that like another
I've heard of exactly one other person who has.
it's weird but effective.
Yeah,
Right? I got it from my mom's family. I mean, she literally had like a freezer. You know, like people have people hunting like big giant freezers. They have that full of like all vacuum sealed recipes.
Yeah, oh dude, I'm
So I did my research.
well, I am.
Yeah.
I mean, you'd say that. Well, now you can never be expert. Just go watch this video and it's like
Magnification is messed up. The only thing that's weird for me is that it seems where the virus is breathing other pathogens on mostly, and he says that he's going to make Frank, he's going to make it really hot. He's going to do sugar, toxins, things like that. Virus. It's like his goal, he's like, I've killed one little girl, kill Frank before 2 hours. Well, I don't got shit on So maybe they need a fictional virus. The back review, now it's your job to make your own review, and then you can go viral and into it. Make sure that one right seat off the bomb. You know, we would just short it. You'd be like, hey, Dr. Off the bomb. I'm like, by the way, my opinion is not next time you want to cancel lecture, don't cancel it for you sit here. It's like an hour instead of Canvas. We're just you'd be on this stage and we wouldn't get you a plaque because that is not very like.
All right, guys. So we will pick up on antibiotics. We've got 31 slides to go. Hopefully we'll make it most of the way through this deck today, I think. And we're finally starting to talk about antibiotics. So we really won't, and we'll talk about antibiotics again a little bit tomorrow when we get into tools and techniques. Not tomorrow, then on Monday. But that's like how we're gauging antibiotic susceptibility. So this is really the only time that we're going to talk about antibiotics in any detail in this course, because this is not a course that extends to treatment options. It's just this appropriate for microbiology because we happen to be talking about the bacteria themselves. So as we're talking about targets, might as well start introducing you to how these antibiotics are working, what they're actually targeting with bacteria. And then very briefly, we might touch upon some antivirals and some antifungals, and even a little bit of antiparasitics, but mostly this is antibiotic oriented. All right, so straight up definition, we're talking about chemicals that can be used to treat bacterial infections. We have a couple different ways of stratifying how they work: bacteriostatic antibiotics and bactericidal antibiotics. With bacteriostatic antibiotics, you're not, strictly speaking, killing those bacteria, you're just inhibiting their growth. Bactericidal antibiotics, your intent is to kill them. So most antibiotics are generally kind of loosely categorized as one or the other. However, that can be dose dependent. So if you crank up the dosage on a bacteriostatic drug, for example, it could become bactericidal, depending on how you use it. For bacteriostatic antibiotics, I can't even think of a reason why it might actually be a good idea to use a bacteriostatic drug instead of something that kills the bacteria. Yeah, what about gram negatives?
They release granules that are bad.
Endotoxin release, yeah, in the LPS, we talked about lipid A and the fact that you have endotoxin activity, which, as long as that bacterial cell is intact, is not a problem because it's integral, right? But when the bacteria die, especially when they die in massive numbers, you get the endotoxic release, and then temporarily you get all those immunopathologies that we talked about. So TLR4 activation of like a cytokine response that gets out of control, inflammatory response. That's called the Jarisch-Herxheimer reaction. Undesirable. Would be better to use bacteriostatic drug in those instances if you know it's a gram negative and if it's an option. With selective toxicity, we're talking about the preference to use antibiotics that, as much as you possibly can, would target something specific to the bacteria themselves and not the host. So that's why we spend, or at least one of the reasons why we spend time talking about bacterial structures, so that you have an understanding of what these antibiotics are actually targeting. Hopefully, they aren't targeting anything that can damage you. The more an antibiotic shares a common target with you, the host, generally you get some pretty nasty side effects or toxicities associated with those antibiotics. So that's what we mean by selective toxicity. And sometimes you have no choice. Some pretty nasty antibiotics, like colistin, for example, nasty antibiotic, lots of toxicity associated with it. But it's a drug of last resort. Sometimes you have to use it. You have no other choice because you're dealing with multi-drug resistant bacteria or something else like that. With bacterial resistance in particular, this is one of the reasons why sometimes we are forced to use combinations of antibiotics. Another reason, with the thinking being that the more antibiotics you pile on an infection, it slows down the resistance rates in those bacterial populations. Another big reason is just because it's part of empiric therapy. I'll circle back in just a second. So when you have no idea what is causing a patient's sickness, let's say they have pneumonia or something like that, you might need to prescribe a couple different broad spectrum antibiotics initially until you get culture and susceptibility results back from the lab, because there are a bunch of gram positives and gram negatives that can cause pneumonia. There are a bunch of bacteria that can cause a bunch of different infections in general. So that's another reason why you might just need to cover your bases initially. Yes, sir.
So is the rationale for an increase in resistance chances because we're using different vectors essentially? So you don't know what it is, or if you're a treatment, you're going to use drug A, which is a static, and drug B, which is a cycle, and then you run cultures, you figure out exactly what it is that's being applied.
Yeah, so the strategies can be pretty complex. It's not necessarily like I want to use a static with a cycle or anything like that, but the end goal is usually I want to wipe out, I want to annihilate whatever is causing this infection. So, like if somebody comes in with meningitis, for example, I've been listening to podcasts with infectious disease docs being interviewed and how they treat patients who are displaying like acute meningitis symptoms and what they're doing. These guys are throwing like 5 different broad classes of antibiotics on their patients. Not just antibiotics, like they're including antivirals too for viral meningitis. So it's because they have no idea what is causing that patient's meningitis, just casting a wide net. They might have more sophisticated strategies, but it just it depends on the presentation oftentimes. Did that answer that question?
Yes.
Okay, all right. Okay, so when you are prescribing combination drugs, for whatever reason, the assumption is that they're going to work well together and synergize, where the combined effect would be better or more effective than if you were to use those drugs solo as monotherapies, but that's not always the case. So when you study antibiotics in detail, starting in like pharmacology, you need to be aware, they'll probably point this out, that not all drugs play well together. Some of them have antagonistic effects. So an antagonistic combination would be an effect where the overall killing effect is lower than if you were to use those antibiotics singly. This is a concept that we'll talk much more about tomorrow or Monday or whenever the next lecture is. Minimum inhibitory concentration of an antibiotic. A simple definition, it is the lowest antibiotic concentration that can inhibit growth of a bacterial population after overnight incubation. So this is not an immediate thing, unfortunately. And you're just looking at an in vitro phenotype. What does in vitro mean? An artificial lab-based scenario. In vitro means glass, but like something you'd observe in a test tube or a microscope slide. It's not in an organism. It's not in vivo. So you get these MICs, and it gives you an indication of whether or not those bacteria would actually be susceptible to the antibiotic that you're considering using to treat that patient. In terms of antibiotic activity, we talked a little bit about the spectrum of activity. Depending on the type, some of these antibiotics can hit more expansive, diverse populations of bacteria than others. Broad spectrum antibiotics like tetracycline, if you look at this long line for tetracycline, it's spanning not just gram-positives and gram-negatives, but it's also hitting some of your atypical bacteria like chlamydias and rickettsias as well. And that's because tetracyclines inhibit, what they're targeting is protein synthesis, specifically the 30S small ribosomal subunit of those ribosomes. So that happens to be effective against lots of different bacteria. And it has a pretty good selective toxicity as well, because what don't we have in our cells, cell ribosomes? Yeah, we don't have a 30S, we have a 40S small ribosomal subunit. So good drug, very expansive, unfortunately lots of resistance sometimes to tetracyclines. And there are some other adverse effects of that. So you contrast a broad spectrum antibiotic like tetracycline to isoniazid, which specifically targets the mycolic acids in mycobacterial cell walls. Is not going to touch gram-positives because they don't have mycolic acids. It's not going to touch gram-negatives for the same reason. Very small, narrow spectrum drug right there. If you knew what you were up against, if you knew specifically that your patient, their respiratory infection was tuberculosis, it would probably be preferable to use a narrow spectrum drug like isoniazid. But again, you don't always have that luxury, and that's why we have empiric therapies. Another nice diagram. A bunch of different antibiotic classes on here. It's just meant to be a nice visual for you to study these different drugs. But if there's a drug on here like clofazimine, for example, that targets DNA replication, we're not going to talk about that. So you don't need to go into any detail studying something on this slide that's not elsewhere in this deck. I probably should have just put white boxes over those to hide those. But in general, this is just a nice slide to study from. If you notice the relative abundance of antibiotics that target cell wall synthesis, that's because it just makes a really nice target for antibiotics. A lot of these tend to be bactericidal. They kill those cells because they lose their structural integrity and their cell wall is destroyed like that. So some really effective drug classes are those that involve destroying or inhibiting or impairing cell wall synthesis. This is straight from the textbook. Another thing that's just to help you study for it, but study for the exam. But to point out really quickly, up here you have some beta-lactams. You don't have to know what cephamycins are, for example. That's actually a subclass of cephalosporins. But most of these other drugs we are going to be hitting briefly. All right, so let's start talking about some of these in a little bit more detail. Start with the cell wall inhibitors, specifically the beta-lactams. So this beta-lactam drug class is a very old class of antibiotics. It was, at least with penicillin, this is the first of this class. It was discovered in 1928, very famously by Alexander Fleming. experiments on Staphylococcus, and accidentally, serendipitously, he dropped or contaminated his Staphylococcus culture with some mold, and this mold was producing penicillin, and it caused inhibition in his Staphylococcus culture. So he was already working on lysozymes, and he was interested in mechanisms to inhibit bacterial growth. So he and another team of bacteriologists in England spent the next decade studying penicillin and its mechanism of action. And they figured out a way to mass produce it, to purify it, and make it available just in time for World War II. So we had penicillin available, certainly mass produced by the end of World War II. And at that time, we really only had one other antibiotic in the arsenal. That was a sulfa drug, which wasn't particularly good. So the beta-lactams as a class, which includes not just the penicillins, but also cephalosporins and carbapenems, what they all have in common is this structural feature called the beta-lactam ring. It is a four-membered, very tightly constrained ring with this mid bond between a carbon and a nitrogen right here, which is important, so remember that bond. And what it does specifically is to bind to proteins in bacterial cytoplasmic membrane called penicillin binding proteins, PBPs. It's a stupid name, but that's what they're called. And essentially what that does is prevent PDPs from doing their job, which is to build that cell wall to contribute to peptidoglycan synthesis. This slide has examples of some specific generic classes of each one of these subclasses of beta-lactams. You don't really at this point need to spend a lot of time studying these, but you're going to get it next semester a lot. So it's good to familiarize. The penicillin class, for example, has methicillin and oxacillin. We've been moving steadily away from natural penicillins into semi-synthetics now to keep ahead of resistance trends. So these are some of the semi-synthetic penicillins. With cephalosporins, we have increased activity against gram-negatives. The penicillins, for example, they do tend to work better in gram-positives. Can anyone imagine or guess why you would have greater efficacy against gram-positives with penicillins? Yeah, they have a ton of peptidoglycan, very thick layer, and they don't have that outer membrane that gram-negatives do. But then why would you have any efficacy at all for gram-negatives with other classes like cephalosporins? Because what? They still have a wall, and that wall still has peptidoglycan, right? In between the inner and the outer membrane, they have that thin stripe of peptidoglycan. So that can be messed up with gram-negatives. And that was sort of the idea with the development of cephalosporins. You want to increase the spectrum to include gram-negatives a little bit more. There have been multiple generations of cephalosporins developed, so you'll hear first generation, second generation, third generation, etc. That's not to keep ahead of resistance as it is to change the range of activity for those different cephalosporins. So like cephalexin, narrow spectrum activity, cephalosporin, cefoxtin, intermediate range, a broader range, and then cefixium has a very broad range. Carbapenems too have been engineered in such a way where these specific R groups allow them to have an expanded spectrum of activity, gram-positives and gram-negatives. If you've never heard of these types of antibiotics like imipenem, meropenem, ertapenem, these are very important types of antibiotics, so you'll definitely hear about those increasingly in our in other courses. And this cartoon is basically depicting in a little bit more detail what we've already talked about. When we're talking about drugs like beta-lactams that inhibit cell wall synthesis. Again, the synthesis is happening here in the cytoplasmic membrane. Regardless if it's gram-positive or gram-negative, you still have the synthesis of those precursors happening here. So as they're being produced, this penicillin binding protein is responsible for facilitating cross-linkage between the peptidoglycan layers and peptidoglycan factor in the cytoplasmic membrane. A couple terms to be familiar with. I do abbreviate peptidoglycan a lot because it's a long word on a slide, just PG. But what we're talking about with the peptidoglycan layer are these cross linkages of these glycans, NAG and NAM, N-acetylglucosamine and N-acetyluramic acid. And these glycans are linked together. Advance the slides so you can see. They're linked together, and then in order to create that really strong, robust mesh-like structure, high integrity cell wall, you have these peptides, pentapeptides, that extend down from those glycans and connect it to the next layer down. So there would be another layer of glycans beneath that in this three-dimensional structure there. Okay. So what those PDPs do are they facilitate the cross-linkage of glycans together in that meshwork. Peptidoglycan layer. Where we can mess that up with a beta-lactam like a penicillin or a cephalosporin or a carbapenem is we have this antibiotic that's coming in from the outside and getting into or past the cell wall into the cytoplasmic membrane. And when it gets into the cytoplasmic membrane, this purple antibiotic here, it's binding to the PDP and preventing it from doing that cross-linking. And when that happens, where it goes wrong for the cell is that you can't fix the peptidoglycan layer, which is a high maintenance structure. So it's not going to destroy an existing layer. That's already intact. But bacteria have to constantly maintain and upkeep that peptidoglycan layer. So this is what is screwed up in that process. It's the maintenance and synthesis. And the other implication there to point out is this definitely affects rapidly replicating bacterial cells more than it does cells that are not dividing. If they're not dividing, they're probably not making peptidoglycan as much. We like using these because they have low toxicity. We don't have any peptidoglycan in our eukaryotic cells, so they're usually pretty safe to use. We pointed out that they're more effective against actively dividing bacteria that are producing a lot of peptidoglycan. And they don't work very well against existing peptidoglycan. So, this cell is just kind of underscoring the point that we do not have that peptidoglycan layer in our cells. Okay. So, unfortunately, resistance to beta-lactams appeared very quickly. We started using drugs like penicillin, and then we had to reflex drugs like methicillin, amoxicillin, and we just kept getting resistant strains. So the problem was these bacteria were producing enzymes called beta-lactamases. What do you think beta-lactamases do?
Break down beta-lactams.
Yeah, they break this beta-lactam ring wide open. So it's an enzyme, it's a bacterial enzyme that's produced, and it cleaves that bond in between the carbon and the nitrogen, that amide bond in the beta-lactam ring. Because of that, those beta-lactamase producing bacteria would wipe out entire classes of antibiotics that we had, important classes. This is a disaster for medical treatment, right? One way we kind of got around that is to prescribe beta-lactams in conjunction with beta-lactamase inhibitors. And that gave us a little bit, it slowed the rate of resistance in bacteria. So we would have these combinations of drugs like ampicillin, sulbactam, piptazo, and other combinations. And that also expanded the spectrum of activity in some of these. So beta-lactams with beta-lactamase inhibitors allowed us to keep using some of these drugs. And we also have other drugs like vancomycin, daptomycin, and bacitracin that also target the cell wall, but they do it in a different way. They're not beta-lactams. And these antimicrobacterial agents, the ones that we use against TB, most of these are first-line treatments for TB. And these two take advantage of the fact that you can target mycolic acids in those microbacterial targets. With vancomycin specifically, we'll talk about this a little bit more when we talk about Staph aureus early in block 3, specifically VRSA, vancomycin-resistant Staph aureus. But the mechanism here is that you have those peptides that join glycan layers together in peptidoglycan. Vancomycin will bind to that last peptide in that pentapeptide chain and it will prevent cross-linking from occurring. Okay, so it's a slightly different mechanism, but that's how vancomycin works. And with bacitracin, this is what this cartoon is showing. It's actually blocking the carrier that moves that prefabricated peptidoglycan from the membrane up into the cell wall. Here we go with those mycobacterial cell wall synthesis drugs. Again, these are mostly first-line antibiotics against mycobacterial cells. Here, this figure is just showing those mycolic acids. This is mostly what would be targeted by these different antibiotics. You could also, I guess, use like beta-lactams too, but I think the penetrating power is really reduced with mycobacterial cells because it's hard for antibiotics to penetrate through this thick layer of mycolic acids. So that's why they tend to have their own drug classes while it hits them. I don't have a nice figure to show the mechanism of action for antibiotics that target bacterial cell membranes, but this is pretty straightforward. We don't have a lot of examples. These tend to be pretty nasty antibiotics. The examples are polymyxin B and colistin. The reason being that these antibiotics, they're trying to selectively target bacterial membranes, but what are our cells encased in? Phospholipid biolayers, right? So there is a lot of toxicity to human cells as well. I put nephrotoxicity here, but there are plenty of other adverse effects that can be caused by drugs like colistin, polymyxin B. So again, usually use these drugs as last resort because of that terrible selective toxicity profile. They work a lot like surfactants we saw in another figure where they just kind of integrate into the membrane. The part that is designed to target bacteria is they do have a preference for LPS, so particularly gram-negatives. They're pretty good at integrating into that outer membrane of gram-negatives. But there is some collateral damage with our own cells as well. And we have some really important antibiotics that inhibit protein synthesis. There's a good acronym for you guys to remember if it's helpful. It's called MALT, like protein in a malt shake, maybe, if that's helpful. So we have macrolides, aminoglycosides, lyncosamides, and tetracyclines. With the first two examples, the macrolides and lyncosamides, I like to study those together because they both target the 50S subunit of the ribosome, slightly different targets and mechanisms of action. But here's another mnemonic device for you. Macrolides, massive, lyncosamides, large, and it's the larger subunit, 50S subunit, so pretty easy to remember. I also have a GIF up here that might be helpful as we're talking through the mechanism of macrolides. So it shows some tRNAs coming in on the right side of this ribosome, and then the polypeptide chain that's growing in the middle of that large ribosomal subunit. And then it's showing the uncharged tRNAs getting kicked off through that ejector site. What was that initial site called? The A site, A for aminoacidocyte. So that's where those tRNAs are coming on. And then that P site, the peptidyl site, is where that growing chain of amino acids is being strung together in polypeptides by enzymes called transpeptidases. So what's happening when you have macrolides, that is binding to the 50S subunit and it's preventing translocation. So you don't have the mRNA being fed through that 50S subunit anymore, and it stops chain, it terminates the chain from growing. Sometimes these are bactericidal, sometimes they're bacteriostatic, I think it depends. But we have some very important significant classes of antibiotics that belong to macrolides. Erythromycin, azithromycin, mentioned before, erythromycin. With lyncosamides, similarly, they also bind to the 50S subunit. They'll block that transpeptidation reaction from occurring, so you don't have those amino acids strung together. And we have aminoglycosides and tetracyclines. These will target the smaller subunit. With aminoglycosides like amikacin and gentamicin, this will cause a misreading of the mRNA as it's being fed through the 30S subunit. Tetracyclines, they have they similarly will block tRNAs from binding when they're coming into that A site. And they have, again, a tendency to have a very broad spectrum of activity. Most bacteria are susceptible to tetracyclines. Then we have inhibitors of nucleic acid synthesis, another table that is out of the Murray textbook that helps you. Quinolones and fluoroquinolones are good for treating bacterial infections, but rifampin, this is more of an antimycobacterial drug, so TB again. And then metronidazole, it can be used for bacteria, but it's also oftentimes used for parasitic infections as well. Mechanism of action for these quinolones and fluoroquinolones is that you are targeting DNA gyrase, which we all know does what?
Lose the engine,
Yes. How does it do that? And why does it do that?
kind of like mix it.
Mix what?
Like DNA.
Further down in the strand, you need to like, so it doesn't get coiled up.
Exactly,
It does,
yeah.
So that it doesn't break under the torsional.
Exactly. So he said that during replication, when you have a bacterial chromosome that's being replicated, double-strand nicks are introduced by DNA gyrase. If you don't have DNA gyrase that's functioning, you can have breakage of the DNA in that chromosome. So that can be bactericidal as well if you don't have these bacterial-specific enzymes working. Because we don't use these enzymes, this tends to be a pretty good mechanism, pretty good approach. We don't have anything like it in ourselves. Well, that's not quite true. We do have topoisomerases, but they're not the same as bacterial ones. So these are additional important classes of antibiotics that target nucleic acid synthesis by interfering with the action of gyrases. Ciprofloxacin is one in particular that you'll probably hear a lot about. For Mycobacterium tuberculosis, again, rifampin is used oftentimes, but because we relied on rifampin so heavily initially, we did get a lot of resistant strains of TB to rifampin, and that's one of the reasons why we always use combination therapy now with tuberculosis cases. Metronidazole is a little bit different. It will also induce breakage in DNA, but it's a prodrug, which means it's not activated until it's in anaerobic conditions, which makes it a little bit safer, generally speaking. So we'll use that for anaerobic bacterial infections, but also Giardia and Ebola infections. With flucytosine, this is a really potent drug, or it can be a potent drug. It's specifically an antifungal and also a prodrug, so it has a safer profile, isn't activated until or unless it gets inside a fungal cell. When it is activated and it's in its active form, it'll disrupt both replication and transcription. There is a problem though, because it does such a good job of messing up DNA and RNA synthesis. It can impact rapidly dividing human cells as well. So this can cause issues in the bone marrow where you're trying to produce leukocytes and lymphocytes. So neutropenia, thrombocytopenia, leukopenia can all result if you're using this particular drug. So this too is one that you wouldn't use unless you absolutely had to for very severe fungal infections, like systemic fungal infections. Okay, nucleoside analogs. These are very, very important, but they are antivirals. Very hard to get good antivirals, so this is a good strategy. A lot of antivirals target viral replication. The thought here is that you have these analogs, these fake nucleosides that will integrate into viral DNA preferentially, so they will outcompete normal nucleosides. And when they do, when they are incorporated into growing DNA or RNA strands, it causes chain termination. So that blocks the virus from being able to continue with its replication cycle. Really important examples here: ribavirin, which is used for some hemorrhagic fever viruses. Zidovudine, that is important for HIV. It's part of AZT, that drug. And acyclovir, very famous drug. You guys know what acyclovir is used for? Herpes, predominantly, and varicella zoster virus. Exactly. And then we have a class that inhibits synthetic pathways, couple classes, and I put these on the same slide deliberately because both of these classes of antibiotics are targeting the same pathway, more or less, just at different points. So here, sulfonamides and trimethoprim are both preventing, ultimately, the goal of making this precursor, tetrahydrofolic acid, which is used to make a couple important things. Thymidine, which is a nucleoside, and purines. What were the two purines we talked about? Adenosine, guanosine, yeah. So 3 of those really important nucleosides are impaired, and then methionine, which is what kind of macromolecule is methionine?
It's a star codon.
It's an amino acid. So the bacteria will have a pathway to make its own methionine, and you can screw that up, and you can screw up its ability to make these important nucleosides as well. With sulfonamides, the mechanism of action is a little bit farther upstream, so it's interfering with PABA and this protein called synthase, interfering with PABA and pyridine becoming dihydroxyuric acid. With trimethoprim, we're working a little bit downstream on dihydrofolic acid, so we don't get the tetrahydrofolic acid, but the same effect, you just don't get the production of these 3 end products there. The reason why this is a good target is that we do not synthesize folic acid, so we don't have this pathway in our body, and that's what gives it that good selective profile. But bacteria, of course, do need this pathway. Okay, almost to the end, guys, and this is a very long lecture. This is a busy slide, but this talks about just a little bit of resistance, and we'll talk about resistance more, but a nice summary of some of the major mechanisms that bacteria develop in order to overcome different types of antibiotics. With these, in general, reduced uptake of bacterial cell, we have porins in that bacterial cell. They can adapt their porins or mutate their porins so that they're more selective in what they uptake, and they can decrease the amount of antibiotics that get into the cell. In order for any of those antibiotics to action what they need to action, they all need to get into the bacterial cell, right? Even if it's a beta-lactam, it still needs to get into the bacterial cell. So all they need to do is make it impossible for them to cross that barrier, and you have some pretty good resistance that develops there. They can also generate efflux pumps. What do you think efflux pumps are? Yep, pretty selective molecules. I don't know how they evolved to have these, but they're managed to selectively pump out whatever antibiotics you're trying to administer to that patient. With target modification, we talked about beta-lactams targeting penicillin binding proteins. With Staph aureus in particular and MRSA, one of the countermeasures that Staph has developed is the ability to produce a different type of PBP called PBB2A, and that's encoded on a gene called the MECA gene. And PBB2A isn't recognized by beta-lactams, so you thwart the activity of antibiotics and targeting normal PBP at that point. And PBB2A, it does the same thing for the bacterial cell. So I don't know if there's much of a trade-off, but that's how you overcome beta-lactams. One of a couple methods. With peptidoglycan precursors like vancomycin, when isolates become resistant to vancomycin, they can change some of the peptides in that peptide that's targeted by vancomycin and become resistant to vancomycin. That's one of the mechanisms that Versa uses to become resistant to vancomycin. Acquisition of a new target, that might be something like that folic acid synthetic pathway. You can use an alternate for that. One of those targets can be changed, and that makes it resistant to trimethoprim or sulfamoxazole. Enzymatic activation, we have beta-lactamases. We've talked about those before, but those two are effective against beta-lactams by cleaving that beta-lactam ring. Acetyltransferases, esterases like that can all inactivate the mechanisms for these various classes of antibiotics. And then again, acquisition of new targets. I think this is redundant. this earlier bullet, but it will bypass the action of that antibiotic. So a lot of stuff, but it is mostly pretty intuitive, I think. Okay, last slide. This is another busy one, but probably a good one to end on, and that's introducing you to escape pathogens. Anybody ever heard of escape before? This acronym? No? Okay, well you probably will in the future at some point hear about this acronym. As microbiologists, we hear about this a lot. We talk about it all the time. But what it does is summarize some of the most concerning multi-drug resistant pathogens that are clinically relevant. These are the pathogens that are acquiring lots of resistance to lots of different types of antibiotics that in turn are causing all these problems associated with antibiotic resistance, transmission of antibiotic resistance genes, treatment failures, increased morbidity and mortality when those treatment failures happen and you have to administer a different antibiotic. When treatment failures happen, that prolongs the stay of patients in hospitals, bumps up healthcare costs, and you get an ever-shrinking menu of options when you have rampant antibiotic resistance. So there are a lot of bacteria that are not on this list that are problematic, and a lot of fungal pathogens, and a lot of parasitic pathogens, and a lot of viruses that have resistance. Like we have a huge resistance problems with Canada right now. But these are the important ones to know, and we will talk about all of these in great detail in Block 3, like the resistance mechanisms and the types of infections they cause. But just kind of be aware of this acronym and watch out for these bacteria. Okay.
Is that like an FYI in the corner, watch out, or?
Nope.
Not good.
No, it's not. No, this is super important. Yeah, you should know this. And that is the last slide of this deck. Third deck is posted, so we'll start talking about tools and techniques tomorrow, which is a super long lecture. I'm sure we will not finish tomorrow. But before we close out this deck, are there any questions? Yes, ma'am. Yeah, like the one much earlier. Yeah, so if I think that's from the textbook, but if there's an antibiotic class that's on there that we didn't talk about, don't worry about it. Yeah. Yes, sir.
What was the whole, kind of unrelated, but what was the whole rationale behind the ivermectin use?
Yeah, so ivermectin is an interesting drug. It was designed and developed with the intent to clear parasitic infections. There's actually the guy who developed it got a Nobel Prize in 2015 for ivermectin. Great drug, right? The thing is, though, you can oftentimes repurpose drugs from their original purpose to something else if they happen to have efficacy, if their mechanism can be extended to something else. And I used to research this in grad school. I was working on antivirals, but some of those antivirals were like hormone therapies as their primary use, or antidepressants, or something totally unrelated. So anyway, it's not outlandish to try and look at a drug like ivermectin and see if it has efficacy against a virus like SARS-CoV-2 that can be used for treatment. The controversy that developed about ivermectin was with regards to whether or not they had done rigorous and robust research on its mechanism of action against that virus. So they were everyone was hopeful and very desperate to come up with good treatments, and I think that was why it was so controversial. They just kind of latched onto it before it had been clinically trialed specifically for those viruses. But it was developed for parasites. Any other questions?
I have one for the right before the escape pathogen slide. The last one, the acquisition of new targets. What is that? You said it was kind of redundant from previous.
Oh, yeah. Okay, so that is the slide that I think that example is referring to is right here. Dihydrofolate reductase. This is an enzyme that's resistant to trimethoprim. So all the bacteria need to do, this particular enzyme is responsible for converting this precursor to that product. If they produce a different enzyme, then they become resistant to trimethoprim. And And that mechanism is by like having a new target. So if you're targeting reductase, just make a different type of reductase and you're good. So
For the overall for the antibiotics, just kind of know their class and like the name mechanism, the mechanism of action for. And be able to identify like this one is this.
kind of, if it's on the, I don't know how to answer that specifically. We'll do a review next week, but general rule applies if it's on the slide, know it. There are things that are stressed during exams and things that aren't, but that's kind of my conservative answer for questions like that. Sorry. Yes, ma'am.
Going back to bacteriostatic versus bactericidal, is the idea of the bacteriostatic is that you just stop the growth and then allow the immune system to push off the
Yes, and thank you for making that point. With bacteriostatic drugs, you want to use them generally on patients with intact immune systems, because that is exactly what you're trying to do. Maybe not intentionally, but when you have an infection, it's kind of a time is an important element, right? Both sides play for time, the bacteria or the pathogen and the human, who can get the upper hand first. The immune system is being aided by that bacteriostatic drug in that it slows down the growth rates of that bacteria and gives the immune system a chance to do its thing, which is evolved in such a way that it takes care of these bacteria in a very tidy, neat way. So that's a great strategy, but you don't always have immune competent hosts either, immune competent patients. So if that's the case, probably better to choose a bactericidal antibiotic. Yeah, that was a great point. Okay, all right guys, thank you. I will see you tomorrow at 10 for trajectory